This is lesson 7 and is continuing to look at movement of substances across cell membranes. This lesson is all about osmosis. This is a special type of diffusion. You have not previously studied this type of movement of substances, and it's relating only to the movement of water molecules. Just like diffusion, substances move from high to low in diffusion. Water molecules are going to move from where they are in high water concentration to where they are in low water concentration through a semi-permeable membrane. So we can see our definition at the top. We can see the water diffusing through a selectively permeable membrane. Remember, selectively permeable, semi-permeable can be used as well, or even partially permeable, I've seen in some books, from a region of high concentration to a region of lower water concentration. So if we look in detail at our diagram, we can see on the left-hand side there are lots of the blue molecules. These are the water molecules, and only one green sugar molecule. So there's high water concentration and low sugar concentration. We say that this solution on the left-hand side is a weak solution or dilute solution. So if you were making up squash, you would have a little bit of squash and you would add lots of water to make dilute or weak squash. On the right-hand side, we have low water concentration, only a few blue water molecules, and more green sugar molecules than on the left-hand side. So we have low water concentration, high sugar concentration. We describe this solution as being a strong or concentrated solution. So again, if we use the analogy of making up squash, if you like your squash strong or concentrated, you put a lot of squash in and very little water. So what happens is our water molecules start to move from where there are lots of them to where there are less of them through the semi-permeable membrane. And as they start to do this, we can see the levels of the liquids change in the two sides of this container. So on the left-hand side, we can see our level going down, and on the right-hand side, we can see our level rising as the water molecules move into the right-hand side. Notice the sugar molecules do not move in this particular example. Osmosis is only about the movement of water molecules, okay? So you would not get any marks for describing movement of sugar in questions, exam questions on osmosis. So we're going to look at examples of osmosis in both plants and animals. So we're going to start with plants first. And osmosis is really important for plants because when water moves into the plant cell, the cell swells and it becomes what we call turgid. Turgid is a particular word we use when the cell becomes firm, when the pressure is increased inside the cell. A little bit like if you're filling up a balloon water bomb and you fill it really, really full, it becomes very, very firm so that when you throw it, it really does explode when it hits the surface that it's intended to. The cell walls are strong enough in a plant cell to withstand this pressure. And it's this pressure that keeps the cell rigid and it provides support and keeps our plants upright. So the stem is upright all the time. If a plant is losing too much water, so on a really hot day, if the plant doesn't get enough water sucked up through the roots and up through the stem and into all the plant cells, then that's why the plant starts to flop over and wilt. Because the cells are no longer swollen and turgid. They become the opposite. The opposite of turgid is flaccid. Flaccid means soft and it makes the cells not so strong and supportive and rigid. So in this diagram here, we can see what happens in osmosis in plant cells when we put them in very, very weak solutions and very, very strong solutions and what they would look like under normal, what we call an isotonic condition. You've probably heard of that word isotonic previously, relating it to sports drinks. An isotonic solution or sports drink is one that has the same amount of water as is in your body cells so that you don't gain any additional water or don't lose any water. So it keeps the water levels in your body pretty much constant, which is really important if you're carrying out sporting activities. So the first diagram here, this left-hand side, shows us a typical plant cell. And we can see the other cells kind of like joining it. But we've got the outside cell wall, the inside cell membrane. We have our chloroplast, we have a couple of sap vacuoles here and a nucleus. And under isotonic conditions, there is no net overall gain of water or loss of water. So we've got 2 arrows coming in and 2 arrows leaving our vacuole. So the water goes into the sap vacuole, but it's pretty much staying the same. It is isotonic. So this would be under normal conditions. However, if you take that same plant cell and you put it in, let's say, pure water, so there's lots more water molecules outside the plant cells than inside in the sap vacuole, because the sap vacuole is a solution. So we call this a hypotonic environment. Water starts to move into the cells, so this time we can see 4 arrows moving into our cells and only 2 arrows coming out. That means that these sap vacuoles are going to gain water, they're going to fill with water, and you get this turgor pressure developing, and the cell becomes what we call turgid. In other words, it becomes really, really firm and solid. The opposite to that is our final diagram here, and just to confuse you, they sound very similar. A hypertonic solution is one where there's very little water outside the cell, and there's more water actually inside the sap vacuole. So in this cell, it would have been put in kind of quite a concentrated or strong, could be a sugar solution, could be a salt solution. And so what happens is water starts to vacate, it starts to leave the vacuoles really, really quickly. So we've got 1, 2, 3, 4 arrows leaving our sap vacuole and only 2 arrows with water coming in. The opposite happens here. We said that the opposite of turgid was flaccid, the cell becomes flaccid. And this is because the cytoplasm literally pulls away from the edge of the cell membrane. It shrinks. This is called plasmolysis. And we can see that the chloroplasts then are kind of squished into this middle part of the cell. And in this particular plant, if all the cells were going through plasmolysis like this and becoming flaccid, then the plant would wilt very, very quickly and become quite floppy and would look as if it was on its last legs and dying. So we need to use the idea of osmosis to help us explain a couple of problem-solving tasks. So this work is on page 37 in your booklet. This is an extended prose type answer. That means that you've got to answer in good written English, you've got to have capital letters, full stops, and you've got to explain using science ideas and terminology why a floppy lettuce will become firm and crisp again if you put it in ice cold water for a while. So if you've got a lettuce that's just on its last legs, it's not dead, it's not going brown, but it's a bit floppy, okay, you put it in some ice cold water in a bowl and it will crisp up again. And the reason this is because the cells inside the lettuce have less water than is outside in the bowl of ice cold water. So the water molecules start to move in from where they're in a high concentration outside the lettuce, through the semi-permeable membrane into the vacuoles of the lettuce cells. Those vacuoles start to swell. The cells in the lettuce start to swell and become turgid, and so the lettuce starts to crisp up again. And these water molecules have moved in by osmosis. So from high water concentration outside to low water concentration inside through the semi-permeable membrane, so the cells become turgid and firm. Another example is we all like to eat strawberries in the summer, and sometimes they can be a little bit hard, they're not quite ripe enough. But if you sprinkle sugar onto a bowl of strawberries and you leave them for about 20 to 30 minutes, after a while you'll come back and you'll see that there's strawberry juice in the bottom of the bowl, and the strawberries will actually soften. So why does this happen, and how can we explain it using the idea of osmosis? Well, if we put sugar outside the strawberry, then that means that the concentration of water is higher inside the sap vacuoles of the strawberry cells than outside, because there's no water in the bowl, it's just sugar. So the water is going to leave the vacuoles of the strawberry cells by osmosis, going from where it's in high concentration of water molecules inside the sap vacuoles to where it's low water concentration outside the strawberries. And as the strawberries lose this water, the cells start to semi-plasmalize. The cytoplasm starts to pull away from the cell membrane, and the cells become flaccid and soft, and so the strawberries become juicier, and that's why we have this liquid coming out, because it's the water coming out of the sap vacuole. So that's the strawberries, the sugar on the strawberries drawing that water out by osmosis. So see if you can write down some answers for those two. And then your last task is we've got 3 pictures here. In the 3 pictures, we've got one picture is showing a plant cell under isotonic conditions. So see if you can guess which number picture is the isotonic one. Look at the arrows to help you, bit of a clue. One picture is showing you a plant cell in a hypotonic solution. So that means there's more water molecules outside, so what's going to happen to that cell? And the final picture is showing you a plant cell in a hypertonic solution. So see if you can match the numbers of these cells to those different types of solutions. And I will go over those now with you. So picture number 2. Number 2 is our isotonic solution. So there's no net gain of water. H2O stands for, is our chemical formula for water. There's no net gain or no net loss of water in this plant cell. In picture number one, we can see the plant cell looks like it's swollen and it's becoming turgid. This is because number 1 has been put into a hypotonic solution. Hypotonic means there's more water molecules outside the plant cell than inside the plant cell. So water starts to move in, the cell swells and becomes turgid. And in the final picture, number 3 has been put into a hypertonic solution. So this could be a strong salt or sugar solution. And there is more water molecules then inside the sap vacuole of the plant cell than outside. So we can see the water molecules leaving the sap vacuole, the cytoplasm pulling away from the edge of the cell membrane, and the cells starting to become what we call plasmalized and becoming soft and flaccid, as opposed to turgid in number 1.